Bismillahirrahmanirrahim. Welcome back to Guytonacademy. Today we are going to discuss our experiment number 7 from the physiology blood practical, clotting time by capillary tube method. And this in this series of videos, we basically discussing the VIVA and OSPE question guide so that you will understand which type of questions can be made from this topic. I hope you will like it. And if you like it, you must comment, subscribe, and like my video so that I know that how much I am successful in delivering my content to you so that it will guide you in your exams. I think there is no video on the internet, especially from Pakistan, for the MBBS, BDS, and nursing and DPT students from the blood practicals. Also, I have made a detailed video on the Power Lab OSPE pattern. I think these will cover all the practicals of first year MBBS. So clotting time. In the case of clotting time, this is a little bit of you can say brief summary. First of all, we will prick the finger, take the blood, a drop of the blood, and then we will touch the capillary tube with our the blood droplet, which will automatically suck into the capillary by capillary action, right? And just after that, wait for 30 seconds. After 30 seconds, break the capillary and note that if the clot is formed or not. In the picture 5, you can see there is a formation of fibrin clot, thread-like structure. This is the endpoint basically. This is the पीछे नहीं जा रहा. हाँ, this is the end point. And finally, when we see the thread, it means this is the time, clotting time, when the clot has been made. So we have to note down this time. And it has a significance in clinical setting and human body. So we will discuss in coming questions. So this is basically the objective that we want to know what will be the clotting time in the human subject. So the normal clotting time is about 2 to 10 minutes if we are using capillary tube method. This is the capillary tube method. First of all, we suck the blood into the capillary by capillary action. Automatically it will enter into it, and then we will break the capillary tube after each 30 seconds and observe if the clot has been formed or not. In this video you can see initially when we break the clot was not formed, but now the clot has been formed. So this is how we observe the clot formation. We need more drier type of thread which indicate closer look for the clotting time. So we have to wait for 30 seconds, and then when we find a thread-like structure, it means a clot has been formed. Now, requirements: capillary tube, lancet, stopwatch, alcohol swab, and cotton. All these are the requirements for this practical. Now, how is the blood collected in the capillary tube? As I told you, first of all, we have to puncture the fingertip by using the lancet. Then, capillary tube is placed at the bleeding site and allow the blood to fill the tube by capillary action. This is how we collect the blood into the capillary tube for the practical of capillary tube clotting time. When do you start counting the time during the experiment? The question arises when the blood is entered into the capillary tube. How long we have to wait? First of all, what you have to do when the blood just enter into the capillary tube, or within 15 seconds, you will start counting the time, or you can start the stopwatch and observe when will the thread-like structure will appear. How do you know when the end point of the clotting time has been reached? The end point is when a fine thread-like fibrin clot forms across the capillary tube. This is a fiber-like structure. So this is the end point when we have to stop the stopwatch and observe the time. What is the normal range for clotting time? Normal range is about 2 to 10 minutes, means depending on the method. Sometimes the puncture is more, so it will take more time. but it will be between 2 to 10 minutes. Define clotting time. Clotting time is basically the time from the onset of injury to the formation of clot, that is fibrin clot. Or you can say from the time of the prick to the formation of the clot is clotting time. What is the significance of determining the clotting time? Means why we perform clotting time. We can perform clotting time for several reasons, like assess the coagulation function, diagnosing the bleeding disorders like hemophilia and liver diseases, monitoring the anticoagulation therapy, evaluate bleeding risk before surgery, evaluating liver function based on the clotting factor production. As you know, lots of the clotting factors are produced by the liver, and identify the hypercoagulable state that may lead to thrombotic disorder. Sometimes there is hypercoagulation, which means the clotting occur very rapidly, which indicate thrombotic disorders. So these all, because of these reasons, we can perform the clotting time. Here I want to mention that I have a complete lecture on the clotting cascade and hemostasis, the mechanism of hemostasis in the body. So you can go to playlist of lectures, class lectures. I think the name is class lectures, and you will find complete clotting cascade lecture on it, so that you will understand complete clotting mechanism. It will be helpful for your practical and theory both. Next question, what is the capillary tube method used for? Capillary tube method is used to measure the clotting time. It is used to assess the coagulation disorders or monitor anticoagulant therapy. But there can be more questions. There can be more question from this topic or any other topic that I have been discussing in complete list of the practicals, because I prepare the questions as much as possible. But maybe because there are several teachers, so they can make more, they can have more ideas and more topics. But I try my best to make as many questions as possible from the topic. It is just like a book, like Firdaus book. You read a lot of things from that. So everything will complete guidance is not in the Firdaus, but where most of the things has been covered in the Firdaus. So similarly, complete practical is not covered in these videos. Here we have covered only the questions related to these topics, so that you can have idea how can we prepare for this practical, uh, what questions can be asked, right? So you can not expect a lot of from this, but at least it will be very beneficial to score very good marks, inshallah. Mention the methods used to determine clotting time. Lee-White clotting time, capillary tube method, prothrombin time, activated partial thromboplastin time, thrombin clotting time. So there are several methods to determine the clotting time, but in laboratory we have mainly capillary tube method, and prothrombin time is also very common. So most of the time we use these methods. What is the role of clotting factors? Clotting factors, are the proteins in the blood that help in the clotting or clot formation. So deficiencies in these factors or proteins can lead to prolonged clotting time. Why does blood not clot in the blood vessel normally? Normally, blood will not clot in the blood vessel because the vessel wall are smooth and preventing the platelets from sticking. Whenever there is a cut in the muscle wall, only then platelet tissue will be exposed and platelet will adhere to the damaged area. Otherwise, if the wall is smooth, then platelets will not come there and blood will not clot. Secondly, because the body release natural anticoagulants like antithrombin and protein C to stop unnecessary clot formation. What initiates the process of clotting? Blood clotting starts with injury to a blood vessel which exposes the underlying tissue and trigger the platelet to stick to the site, as I told you before. Name any two anticoagulants, warfarin and heparin. What are the anticoagulants and how do they affect the clotting time? Anticoagulants are the medications that slow down the process of blood clotting. This will lead to prolonged clotting time. What are the naturally occurring anticoagulants in the human body? Like antithrombin 3, protein C, protein S, heparin, tissue factor pathway inhibitors, nitric oxide, prostacyclin, thrombomodulin and plasminogen. बेसिकली जो लिस्ट है ये है सबसे ज्यादा एंटीकोगुलेंट कौन सा होता है बॉडी में, ठीक है, ड्रॉप डाउन लाइक एंटीथ्रोम्बिन 3 सबसे ज्यादा होता है, फिर प्रोटीन सी, देन प्रोटीन एस, देन हेपेरिन, देन टिश्यू फैक्टर पाथवे इनहिबिटर, देन नाइट्रिक ऑक्साइड. तो इस तरह से सबसे ज्यादा जो है स्लोली जो है वो अमाउंट का कम होता जाता है. तो एंटीथ्रोम्बिन 3 जो है सबसे ज्यादा क्वांटिटी में बॉडी में मौजूद होता है फॉर एंटीकोएगुलेशन. Which anticoagulant generally used in the laboratory? EDTA, sodium citrate, heparin, oxalate and sodium fluoride. ये सारे एंटीकोएगुलेंट हम यूज करते हैं लैब के अंदर टेस्ट करने के लिए. What is the role of calcium ion in the clotting process? You know, calcium are essential for activating several clotting factors that lead to the formation of fibrin clot. So the calcium ions are vital for nearly every step of the coagulation process, and their presence is critical for effective hemostasis. Why do we use capillary tube method in clinics? Because to monitor the coagulation disorders or the effectiveness of anticoagulant therapy, to check if the anticoagulant are working in the human body or not working, so we can change different medications. What factors can prolong the clotting time? Like low platelet count, because platelets are very essential for clotting. Vitamin K deficiency, it can prolong the clotting time. Factor 1, 2, 5, 7, 10 deficiency. Coagulation factor deficiencies, anticoagulant medications or liver disease can prolong the clotting time. What does a shorter clotting time indicate? A shorter indicate hyper coagulable state and increasing the risk of blood clot formation. And this can be due to several reasons such as genetic disorders, vitamin K excess, medications, chronic inflammation and cancer. In all these conditions, there can be faster clotting. For a more comprehensive evaluation of coagulation disorder, what additional tests might be necessary? Prothrombin time, activated partial prothrombin time. These tests are very commonly used, especially in the kidney patients, and these are very common tests other than the capillary tube method. What is the physiological role of platelets in blood clotting? Platelets form platelet plug at the site of injury and activate many clotting factors to initiate the clotting cascade. And this is the primary hemostasis. Initially, it will stop the bleeding. What should be done with used lancets and capillary tubes? Used lancets and capillary tubes should be disposed of in a biohazard container to prevent contamination and injury. What medical conditions can affect clotting time? The conditions that interfere with normal blood clotting process include hemophilia, liver disease, and vitamin K deficiency. What are the key steps in the coagulation cascade? Means normal coagulation cascade. Vasoconstriction, platelet plug formation, activation of clotting factors, and finally we will have a fibrin formation which stabilizes the clot. How does fibrinogen convert to fibrin and why is this important? Fibrinogen is converted to fibrin by the enzyme thrombin during the coagulation process. Fibrin formation leads to the stable blood clot. How do endothelial cells regulate blood clotting? endothelial cells release substances such as nitric oxide and prostacyclin, which inhibit the platelet aggregation and prevent unwanted clot formation under normal condition. However, injured endothelial cells produce tissue factor to initiate extrinsic clotting pathway. What is the function of vitamin K in coagulation process? Vitamin K is necessary for the synthesis of several clotting factors including prothrombin, factor 2 and factor 7, 9 and 10. How does the body prevent excessive clot formation, thrombosis? The body regulates clot formation through natural anticoagulant like antithrombin, protein C and protein S, which limit the activity of clotting factors and prevent excessive clotting. How does thrombin contribute to both clot formation and clot regulation? Basically, thrombin converts the fibrinogen into fibrin, which is the active form, to form the clot. But it also activates the protein C, which in turn inhibits further clotting by degrading the clotting factor 5 and factor 8A, helping to regulate the process. Means on one side, it is activating the fibrinogen, converting the fibrinogen to fibrin to make a clot. However, to control a limited clotting, it will activate vitamin C, which in turn degrade vitamin 5 and 8A, which are also important for the clotting, so that when there is no 5A and 8A, clotting will be stopped. So this is how it will regulate the clot formation. Thank you so much. This is about the clotting time.